All right. Thank you, Giovanni. Nice to be here this morning. And as he said, I'm living more or less in the Kansas City area, so I'm not threatened by the hurricanes right now, which is a good thing. And as we've mentioned, I can move back and forth between hurricanes and tornadoes. So it's kind of interesting for both of those different things. But let me go ahead and get started. Let's talk about today's work. I'm going to bring you back in time a little bit over the last 10 years and talk about the Deepwater Horizon Oil Spill in the northern Gulf of Mexico. I'll give you a broad overview of the research that I've done there in salt marshes. And this is a pretty good graphic to start with because it shows the location of the Deepwater Horizon oil platform about 70 kilometers off the shore of Louisiana in the mouth of the Mississippi River. In gray, it also shows the extent of oiling in the Gulf itself, surface slicks during the summer of 2010. But more germane to my talk, it shows the extent of shoreline oiling in red all the way from Louisiana to Florida. So a couple of details to remind you about the spill. First of all, the blowout of the platform happened in April 2010 that released methane and crude oil into the Gulf of Mexico directly for 87 days. Over 200 million gallons of oil gushed from the well during that period of time, and it was the largest accidental release in history. Picture up in the lower upper right just shows the fire on the platform where 11 workers died as a result of the explosion and fire, and a couple of pictures of the oil slicks on the Gulf of Mexico. Been a lot of research done on the continental slope, especially by Paul Montagna for Meiofauna and Patrick Schwing for foraminiferans. But I want to move inshore. I want to talk about the inshore oiling that happened, the shoreline oiling. It was also the most oil in history that reached the shorelines in the northern Gulf. About 2,100 kilometers of shorelines were oiled. And my specific focus will be about the 1,000 kilometers of coastal wetlands that were oiled. So when this spill happened, given the ecosystem services provided by the coastal wetlands in the northern Gulf, certainly was an unprecedented potential for environmental damage and was a great concern. So just to give you a sense of what this oiling looked like in these salt marshes that I'm going to talk about, I'll show you a picture from the air here. The marshes were oiled often heavily to about 10 meters inland from the shoreline edge, and you can see from this picture from the air this black band associated with the shoreline itself, and that's all oiling. It's oiling that happened in that first few meters. The oil itself was weathered. That means that the lower molecular weight fractions had evaporated. It was heavy, viscous, oily residue, and it really smothered the plants and invertebrates in those places which were heavily oiled, and the oil was even incorporated into the soil structure. So I like this picture down below. It shows standing from the shoreline looking into the marsh platform. You can see this band of heavy oil residue as well as dead plants associated with it. So this is the kind of environment which I'll be talking about today. So given that possibility of such a damaging situation, a group led initially by Irv Mendelson at Louisiana State University began conducting investigations to look at the responses of vegetation, benthos, and microbes, including the microalgae. So we coordinated those efforts. A lot of participants have come and gone through the 10 years after the spill, but here's a group of one listing of some of those participants. You might recognize Kevin Carmen's name. I do want to recognize, though, Rita Riggio and Manisha Pant. They sorted and processed most of the rough sorting of the samples that I'll talk about today, so they get special recognition for the efforts that they put forth. So I like to think of this as an ecosystem approach, and the reason for that is that they were coordinated studies that looked at vegetation. I also looked at the benthic microalgae and the Meiofauna and the Macroinfauna. So a couple of pictures of our group out here working at reference sites in that northern Gulf and the shoreline edge. So I'll try to indicate some of all these different factors and try to integrate them together and how they influence the Meiofauna. To make this work, we had to have some sampling locations. We decided on 7 reference sites that I'll abbreviate RF, 7 moderately oiled sites as MD, and 7 heavily oiled sites I'll abbreviate as HV. All of those were within 3 meters of that marsh edge, and the sampling areas were located over a location of about 8 by 5 kilometers. So essentially what we did was to take samples every 6 months Some samples were collected as early as 9 months, some after the spill, and some as long as 9 years after the spill. Specifically for the Meiofauna, I'll be talking about samples from about 18 months after the spill to about 6.5 years. And I do want to show you the location of our efforts. Here's a map of Louisiana. About 95% of the oiling, 90% of the oiling and shoreline happened in Louisiana. The red box is illustrated here. That area is called as Barataria Bay. It's just west of the Mississippi River. And this box here indicates where all of our sampling was done. And that red zone, that's where the heavy oiling took place. What happened was the wind blew the oil into the Barataria Bay and continued to blow it northward until it came ashore in those areas that are designated there. So maybe I should first, I'm going to say the word recovery about 10,000 times today. So maybe I should define what I mean by that. First of all, I want to say that there were no before data from those exact locations, except for vegetation, which is helpful, but doesn't help much with the Meiofauna. However, we have lots of sites from across the region that were unoiled, and we have those reference sites. So what I did was to compare the densities and community parameters at oiled sites with those from the reference sites, those RF sites, which were companion sites, as well as the numerous unoiled sites across the region. So I'll define recovery as the time at which there was no difference between the oiled sites and the reference sites. And I want to indicate that I'll really focus on the heavily oiled sites. And the reason for that is there was such a severe disruption of the ecosystem functions and processes and services that seems to need special attention. Essentially, the plants, the microalgae, the benthic invertebrates were wiped out in those areas or really greatly decreased in abundance. And in that sense, recovery is quite special. It really resembles secondary succession or the process after which salt marshes are restored, a sort of habitat maturation process. Now, I don't have time to talk so much about the moderately oiled sites. Effects were milder, they were still long-lived, but I'll try to focus on the heavily oiled sites because of the severity of the effects in those locations. So I'll divide up the presentation into 3 subsections. I'll talk about the early stage recovery of the plants and the algae and the infauna. That would be less than, say, 2 years. Then I have a time from 2 to 6.5 years where we did some intensive investigation to look at the factors that promoted recovery. And then a novel part of the study, that's the part that's not yet published, has to do with the macroinfauna. And what we think is happening in this case is that the macroinfauna, as bioturbating ecosystem engineers, may have facilitated some of the recovery of the Meiofauna, which is a new and kind of interesting finding. So I'll break up the talk into those 3 sections. First, I want to mention the initial recovery period. And to do that, I need to say something about the plants in the area. The marsh macrophytes in salt marshes act as foundation species. They make life more possible for the invertebrates which live there. And in the northern Gulf of Mexico, really there are 2 species that are co-dominants. Spartina alterniflora would be the most abundant, the smooth cordgrass. But secondly, Juncus romerianus, the black needle rush, is also a dominant species as well. And their response to the oil spill was quite different. The other thing I want to mention is that in these intertidal settings that become air exposed at low tide, really kind of consider them as a soil and not so much a sediment. And as such, the roots and rhizomes of the plants that grow there are really significant contributors to the environment. So I want to say those kinds of things because we'll mention that throughout the talk as well. So first I want to mention Spartina and Juncus. Spartina is the main species in that area, but it was much more tolerant of oil and it began to recover much more quickly in those heavily oiled sites than Juncus. Juncus is less tolerant and its recovery has been much, much slower. But an important point is that neither the above ground biomass nor that below ground biomass of living roots and rhizomes recovered over the 9 years of the study. So there are long lasting effects on the plants throughout this environment. So what do I mean by that? Well, here's a great picture, right? It shows the same site 9 months, 18 months, 30 months, and 42 months after the spill. And you can see from the site itself at 9 months after the spill, there were essentially no plant life present. By 18 months, it was starting to return. By 30 months, there was definitely a return of Spartina. And 42 months, there was quite a bit of Spartina growing there as well. So that's great news, right? The plants did start to recover after a pretty quick period of time, say 18 months. But I do want to say that even after 42 months or even after 9 years, the full recovery of the vegetation had not taken place. And this had ecosystem level consequences to the benthic community. One of the reasons for that is that over time, the vegetation cover remained reduced in those heavily oiled marshes. There wasn't as much plant life there to cover the soil, and light that reached the soil sediment was increased, and we think that affected the benthic microalgal biomass, because overall there was a lot of algae in those heavily oiled sites and occasionally spiked up very high biomasses. And just wanted to show you this picture at 30 months again. You can see all the gaps. There's lots of plants, but there's plenty of gaps in between them where light could reach the surface of the sediment. So the plant life was driving a lot of this, and one of the effects they had was an effect on the benthic microalgae. So let me talk about that next. Again, within those heavily oiled sites, there was essentially 100% mortality at the beginning of the spill itself. But interestingly enough, the biomass and pigment composition recovered during the second year after the spill before the meiofauna recovered. So there was a pretty rapid recovery, at least in terms of the pigment composition and biomass over time. So let me show you a figure that represents that. This is the chlorophyll A concentration in the sediments, which is a proxy for the benthic microalgal biomass. The way this figure works, and some subsequent figures work, will be that the x-axis will have the years after the spill, or maybe the months after the spill. I'll always have a green bar for reference sites, a yellow bar for moderately oiled, and a black bar for the heavily oiled sites. The other thing I've done is to add some dashed lines to represent regional averages, so you can see what you might expect for the region in terms of chlorophyll A concentration. So even after the first year, during the second year, essentially the differences between the reference moderately oiled and heavily oiled in terms of chlorophyll A had pretty much vanished. So there was a really rapid recovery, and they stayed very similar in time throughout, except for the fact there were a couple of instances where there was real algal blooms associated with the heavily oiled conditions. So here's one at 30 months, and you can see that there was a higher than the regional average for the benthic microalgae at 30 months. And there was actually a very prolonged increase in benthic microalgae from about 5 to 6.5 years. I put them in different colors because I'll refer back to these two different spikes just a little bit within the talk itself. So benthic microalgae recovered quickly, and it responded in different ways. It responded by having a lot of algae in some of the heavily oiled sites. Okay, so what about the meiofauna? Well, I'll define the meiofauna here as the animal community that passed through the 0.5 millimeter sieve, and they were retained in this case on a 32 micrometer sieve. And again, in those heavily oiled sites, there was massive initial effects on abundance and diversity. I just want to show you one taxon as a representative example of that, and I've chosen copepods, partly because I've worked with copepods a lot and they're my favorite group, but that's okay. And what that shows is, first of all, that even at 18 months, even as things were starting to recover, there were still very low densities in the heavily oiled areas, heavily oiled sites. But by 30 months or 36 months, there really were no differences in the copepod density between the heavily oiled, moderately oiled, and reference sites, and that remained over time. There were what might be seasonal variation. They go up and down some, but essentially by 2-3 years, there was a recovery of the copepod abundances. I did the same thing for copepod species richness. It followed the same pattern. The numbers of species had a strong initial decline, and then they were followed by recovery after about 2 years. The other thing I want to mention is that that spike in benthic chlorophyll A seems to be represented with a spike in the copepod abundance at 30 months at the heavily oiled sites, and that's what that circle is supposed to represent. Turns out there was a strong single species bloom in that heavily oiled conditions only at that period of time, and it was certainly coincident with the microalgal biomass that was present in those heavily oiled sites. Species was in the genus Harpacticus. It bloomed way above the normal background density. It made up 43% of the collection at HV, but only 1% or less at the other sites. And certainly indicates that the community as a whole was still disturbed, and they didn't recover. The community doesn't recover as quickly as the density does for some of these individual taxa. All right, The other thing then that I have is a strong database from 2 years to 6.5 years that have maybe allowed me to look at the factors that could influence the recovery. So I used this period of time as a more intensive investigation to see what organisms were responding, how they responded, and how they recovered. To do that, I used the what are called effects ratios or response ratios. And that's just a simple, straightforward way of saying that I took the average density at the heavily oiled sites and divided by the average density at the reference sites for the same period, and then I plotted the ratios against time, over time after the spill. So essentially what we see then is a dividing point. The ratio is less than, if the ratio is less than 1, it suggests there's higher densities at the reference sites, and therefore the reference hasn't become, the heavily oiled sites haven't become equivalent to the density at the reference sites. And I have a lot of values where there's higher densities at the heavily oiled sites than at the reference sites. So those values would be a ratio greater than 1. So you can see that on each one of these figures, I've got a dashed line to indicate that ratio of 1. Now, what I did was to group the animal groups into 3 different figures here for you because they acted quite differently from each other. There was a group of Meiofauna that recovered very quickly. That consisted of copepods and nematodes, and annelids excluding one species called Meiobenthos estuarina, which is a sabellid tube-building polychaete. So if I look at those groups and look at the time course, they're essentially recovering in about 2 years, maybe 3 years. So some groups of animals recovered very, very quickly: nematodes, copepods, and annelids except Meiobenthos. Interestingly enough, there was a second group that also recovered. They reached that magic value of 1 at some time during the study, but later and slower. So actinaids, amphipods, and bivalves, juveniles, all of these recovered more slowly, but they did recover in, say, 3 years, 4 years, or 5 years. The other thing about them is that if you look at the ratios, the abundances were really super high at the heavily oiled sites. In other words, the ratios were greater than 1. They were sometimes 4, sometimes 6 times more abundances at the heavily oiled site than the reference sites for those taxa. So these taxa recovered more slowly, but they did reach and sustain very high densities in the heavily oiled sites for later on in the recovery period. And then thirdly, there was a group of animals that just weren't resilient. They didn't bounce back. And that includes a species of chinorhynch. That includes this sabellid polychaete called Meiobenthos and juvenile gastropods and ostracods. And if you see their abundances never reached that value of 1, that ratio of 1. So we have 3 different groups. So it's kind of a great situation to be able to ask what factors were important to these different organisms. Why did some recover fast, some more slowly? And why did some not recover? And can we say something about that? So I looked at the factors that might drive the recovery through the community itself. So I defined the Meiofauna communities at reference, moderately oiled and heavily oiled. And then I looked at them for that 4.5-year period starting 2 years after the spill. So I defined the community in terms of 12 different taxa. Some of them were temporary Meiofauna, some permanent. So we had juvenile gastropods, for example, and juvenile bivalves. But then we also had nematodes, copepods, ostracods. These are the most abundant of the Meiofauna taxa associated with this. And I did a couple of individual species, Meiobenthos, the Tanaid species, and a chinorhynch species. So it was a mix of different taxa. But then because we're coordinating these studies, I had a lot of information that was accessory to that to give me a sense of what the environment was doing. So I divided up the 8 factors that I looked at, and some were having to do with foundation species. So we looked at the Spartina and Juncus, the above ground biomass, that would be the weight of the stems and leaves above ground. Also looked at the benthic microalgal biomass because that's important as a food source. And then the soil quality was also important. And I used measures like live below ground plant biomass, that would be the roots and the rhizomes, the pH of the soil, density of the soil, and the hydrocarbon concentration. So I did a community analysis. I used a distance-based linear model in Permanova as the approach. What this program does is it creates a model that estimates the relative contribution of each of the environmental factors I looked at and how much of the community variation each of those factors explains. So essentially this program and this technique develops a model which lets that be done. So I want to show you the results of the model from that 2 to 6.5 year period. I used 8 different environmental factors. 7 factors contributed to the model. The only factor that didn't was pH. pH did not contribute to the model. Some of these were significant contributors, others were contributors but not significant. And I want to mention just a couple of things about this slide. One is that overall, these 7 factors explained about 27% of the variation in the meiofauna community, which I thought was pretty reasonable. And 2 factors contributed most of that variation. One was the above ground biomass of the plants, Spartina, and the other was the microalgal biomass. They both contributed about 7–8% of the variation to explain that in the meiofauna variation. These things kind of make sense as factors that will be driving the recovery. So as foundation species, Spartina has a great effect on the physical structure, the production, habitat cover, and so forth of these areas. And essentially, above ground structure accelerated that initial colonization and long-term recovery. So Spartina was definitely a factor throughout time. The other factor was the benthic microalgae, and as I've said before, there were instances when it was quite high, and that certainly serves as food. So the microalgae were recovering in the second year of the post-spill. Spartina was certainly recovering, but not fully after 2 to 3 years. And a lot of animals either recovered simultaneously or within a couple of years thereafter. So these are positive correlations that remain through the study period. So some animals, those especially that recovered at all, recovered quickly, they responded to algae and they responded to above ground structure. But what about those 4 taxa that didn't recover? What were they responding to? Well, I used DISTLM to try to get a sense of that as well. I don't have time to show you the results of the analysis, which I did for this period of time. But let me do say that marsh soils are really important to the recovery of sediment-dwelling animals after habitat restoration. So when marshes have been restored, people have looked at the marsh soils and the infauna and find that marsh soils are very important to that end. So 4 taxa didn't recover at the heavily oiled site in association with factors such as below ground biomass and such. So let me show you a couple of indications of that. One indication is this is just a plot of the below ground biomass, again, the live roots and rhizomes over the 6-year period. And the important point to get at would be that it's easy to see that the heavily oiled sites just never recovered. We continued this study for 9 years and we found the same result, and that's been shown in other restoration related studies when marshes have been rebuilt. The below ground biomass can take decades for it to recover after restoration, and now we're seeing after an oil spill as well. How was that manifested? Well, 2 taxa that I want to mention. One would be Chinodaries, Cooli, the Chinorite, and the other is Maniokia estuarina, a 2-building polychaete. You can see those abundances were quite low during the all through time, and especially in the heavily oiled sites, they just never recovered at all. So these things were slow to recover, in part because the below ground structure of the marshes was slow to recover. Why do we know that? Well, lots of studies have been done on wetlands and terrestrial systems, and in terms of how systems respond in terms of ecosystem functions, it's very important to realize that these below ground factors are quite important to that. So live below ground biomass and bulk densities are good indicators, and they affect lots of different things within the soil, like the cohesiveness of the soil, and they could affect burrowing and tube building and so forth. So certain factors then seem to slowly recover, and some fauna were responding to those factors quite differently than the other fauna. So some recovered and some didn't recover over that period of time. All right, last but not least, then I want to talk a little bit about macroinfauna, because this is a rather novel part of the study, and I think I was uniquely positioned to be able to say something about that. I'll define the macroinfauna as those animals which were retained on the 0.5 millimeter sieve, and since I did the macrofauna and the meiofauna from the same samples, I can compare them. So I'm going to look at the macrofaunal responses compared to the meiofauna responses from the same samples. So these, of course, are the larger animals, the polychaetes and bivalves and so forth, which live in the sediment. And let me just start by saying there was essentially no macrofauna at those heavily oiled sites until about 2 years after the spill. However, I do think macroinfauna have a great potential to influence the meiofauna. They're usually overlooked as an important environmental factor for the meiofauna, but in general terms, they could be, especially after an oil spill. We know from many studies after oil spills that the species composition of the macroinfauna changes over time. Often the species will reach very high densities and abundances in particular phases after recovery. It's almost like a successional event where some species of macroinfauna come and go. And certainly then that gives the potential for great effects on the meiofauna. The reason for that is that the meiofauna could be influenced by those macroinfauna. So I want to ask questions about how the macroinfauna that change over time could affect the recovery of the meiofauna. And there are a lot of ways that those interactions could occur. The macroinfauna, of course, are much larger than the meiofauna. They can occur in very high densities, so they may outcompete the meiofauna for food. Predation on the meiofauna is possible, but a big influence would be bioturbation. A lot of macroinfauna are called ecosystem engineers because they have such a great influence on the sedimentary and soil environment, and they can influence that indirectly in a way that indirectly influences the meiofauna. So just let me mention bioturbation here. The macroinfauna do a lot of things to alter the sedimentary environment. They irrigate burrows like is shown in the lower left-hand figure here. They move sediment around, they defecate, they alter the soil chemistry, oxygenate it, for example, and they increase the particle resuspension. That's what I hope to show you in this right-hand side of the figure. Here's a worm that actually increases what's going up into the water column as well as altering the sedimentary environment itself. Lots of studies have been done now to show there are really strong species-specific effects of the macroinfauna on the meiofauna. Certainly the density, distribution, and diversity and density and distribution can be affected. So what's going on with the macrofauna after the spill? Can we say something about that? What I want to focus on are the amphipods. They were the most abundant group of the macroinfauna after about 3-4 years from the spill. And interestingly enough, one of the species which is present and dominant is potentially a bioturbating ecosystem engineer. So I want to try to illustrate what could have happened with that and what we think was going on. Now, a lot of studies have been done on a very important ecosystem engineer called Corophium volutator. It's a species in Europe and North America. In estuaries, it's been studied extensively. We don't have that species in Louisiana salt marshes, but we have a very close relative called Apocorophium louisiana. That species made up over 90% of the amphipods in our studies, and they're very closely related, have very similar morphologies and so forth. So a large number of studies have been done on Corophium volutator, and that suggests within the family and subfamily of Corophium, there's a certain similar morphology, certain similar behavior, and certain similar effects. So we're assuming then that Apocorophium acts a lot like Corophium. But what this group of organisms has been shown to do is that they're surface deposit feeders, and they seek out and consume benthic microalgae. In fact, they reduce the abundance of benthic microalgae. They live in very highly aggregated groupings. They move as metapopulations with high rates of dispersal for adults and juveniles on the scale of tens of meters. In other words, they can decide where they want to live, and they move from place to place, and they follow the benthic microalgae, at least based on what we can know about that. And they're also exceptionally capable bioturbating organisms. They irrigate their burrows, they move particles around, they move the nutrients around, and they irrigate their burrows and thus send things out of the soil into the water column. Just a couple of pictures of Corophium. This is a textbook representation of Corophium as a surface feeding copepod, moving currents of water in and out. It's not a very accurate representation because we usually think of the tubes that Corophium build as U-shaped. So here's a drawing of Corophium at the mouth of a burrow, and here's a picture which shows that really well. Here's the U-shaped burrow that Corophium lives in. Here's the animal in the burrow itself, and of course what it's doing is irrigating that burrow and feeding on the microalgae that it stirs up as a result of that. And just one more picture of Corophium in the burrow itself. So what happened with these macroinfaunal amphipods after the spill? So this now is density in per meter square, so it's the animals which were retained on the half millimeter sieve. As I said, there were essentially no animals for the first 2 years after the spill. They gradually started to recover from 2 to 4 years, 2 to 3 years. The recovery actually happened first in the heavily oiled sites, and the density recovered in about 4.5 years. However, that's really kind of interesting that the densities really went very, very high after the spill itself, after that recovery period in those heavily oiled sites. And in fact, here's that representation of the benthic microalgal biomass, which was really quite high from 5 to 6 years. and there was a density peak coincident with that. So the density of Meiofapods really correlated with benthic microalgae. So that seemed to be part of what was going on here. The other thing about it is, if you noticed, that the densities were much higher than the regional means at the heavily oiled sites. The regional average is about 3,000 per meter square, and we had densities of 60,000 per meter square on average at the heavily oiled sites for periods of time. Interestingly enough, there were more amphipods than most of the Meiofauna taxa. There were more amphipods in the cores than there were than all the Meiofauna taxa except nematodes and copepods and Meiounkia. And what surprised me as a copepod person, there were more amphipods than copepods in about 10% of the samples. So think about that for a minute. These would be juvenile and adult amphipods. There were more amphipods than copepods in about 10% of the samples. So I think it's a unique situation that I wanted to exploit a little bit. So what happened was amphipods disappeared at heavily oiled sites. They were extirpated. Then the recovery began about 3 years later. The density was recovering certainly by 4.5 years, and then there was this big surge in abundance afterwards from 4.5 to 9 years. But the Meiofauna weren't fully recovered from the spill during this period. Some were recovering while that increase took place, and then some were never recovered at all. So to me, it's like a natural experiment. What I wanted to do was to try to ask questions about this amphipod abundance to see if the amphipods would affect the recovery of the Meiofauna. So I'll do it in two ways, right? I want to ask if the amphipods influenced the recovery of the Meiofauna that were slowly recovering, like Tanaids. They did recover during that period of time, but they were also recovering while the amphipods were really abundant. Then I wanted to know if amphipods could slow down the secondarily affect the Meiofauna that did not recover over that period of time. So I think it's an interesting question. It's novel. Not many people have looked at this in the past. And if you looked at past studies on Corophium volutator, they're kind of mixed results. Some studies have shown either positive or negative effects on the Meiofauna. So I didn't really know what to expect from this, if there would be an effect or not. So I used a very simple approach. I used correlation analysis over that period of time, over that 2.5 period of time, and I used the densities from the reference sites, moderately oiled and heavily oiled. So there's a big range in densities of amphipods. And I correlated the density of the amphipods with the density of the various Meiofauna group from the same samples. So I had 72 samples in which I was able to do that. So how can I portray that? Well, what I decided to do and what I did first was simply graph everything out. So I graphed the density of amphipods on the x-axis, and then against the density of various Meiofaunal groups on the y-axis. And what I've done here is to place the species, the taxa responses where there was a positive response. In other words, for all these taxa, there was a very distinct positive result where there were more amphipods, there were more of these Meiofaunal groups. So for copepods, there was actually a very high R, correlation coefficient of about 0.65. For Nauplii, it was even higher, 0.675, and then the highest correlation was for Tanaids at about 0.8. So really interesting things. These are the taxa that recovered in 6.5 years or less. All of these groups, the juvenile bivalves and so forth, they all recovered in about 6.5 years or less. And for all of these, the abundances were positively correlated with amphipod abundance. Very interesting, isn't it? But remember, not everything recovered, and what I've done here is to plot the Meiofauna that didn't recover. That was Meiounkia, Chinorhynchs, and so forth. For those organisms, the correlation coefficient was quite low with amphipod abundance. It was only negative 0.17 for Meiounkia. For Chinorhynchs, it was 0.06. None of these values were significant, so there was no relationship, no correlations with amphipod abundance. And these were the taxa that didn't recover after 6.5 years. So they didn't seem to care about the abundance of the amphipods. They were not recovering, and the effect of the amphipods doesn't seem to be there. There was no effect. So if I can summarize that, there were no negative correlations with amphipod density with any of the Meiofauna. Even those very high densities, 20 times higher than the regional averages, amphipods didn't slow down the recovery of Meiofauna. They didn't alter or slow it down in any way. Rather, for many taxa, they responded positively to the high densities of amphipods. Those taxa are listed here, and again, Tanaids and bivalves, they were recovering while the high densities of the amphipods were present. Now, one thing that seems to link all these groups together is they tend to consume benthic microalgae. So I can kind of pose two hypotheses, I think, or two possible reasons for this. One is that the meiofauna and the amphipods responded to the same factors, and a good one might well be benthic microalgae. So in other words, then I could ask another question, and that is, did the density of the meiofauna correlate with benthic microalgae? And maybe they're just, maybe the meiofauna and the amphipods are responding to just the presence of benthic microalgae. But there's another possibility that's been followed through studies of Corophium volutator, mostly in Europe, and they've considered lab and field studies. And the possibility there is that the amphipods are facilitating the recovery of the meiofauna. By that I mean some studies suggest that amphipods can stimulate benthic microalgal production and make more algae present, more algae available. And also, when bioturbation takes place, that increases the suspension of surface of the microalgae into the near bottom water, and that could make more algae available to animals like copepods and some polychaetes that feed right in the surface of the sediments themselves. I show you here the correlation coefficients between chlorophyll A concentration and the density of various meiofauna groups. And what I've done is put all the ones that were significant on the right and put them in blue. And all of these groups were groups that were significantly correlated between chlorophyll A and meiofauna. And these, again, are the same groups that responded positively to the amphipod density, which was present there as well. So it seems likely then that attraction is a possible explanation. In other words, amphipods were positively associated with chlorophyll A, as were many of these meiofauna groups. And these are the groups that recovered. The other groups that didn't recover don't seem to care about chlorophyll A. They didn't care about amphipods as well. However, so it's possible that there was just an attraction, but facilitation, I think, is also possible as well. And I want to use a couple of studies that have been done to illustrate that. One is a study by Weirman that I mentioned down below, and this figure that you see here came from that paper. And what they did in this paper was look at differences in grazer's abundance, that's the abundance of Corophium, and measured the primary production of the benthic microalgae. And what they found was that the more Corophium, the higher the primary production of benthic microalgae. So clearly, if there's more production, that might be a facilitation that could occur for the meiofauna. The other thing that's illustrated here is the possibility that there's more particles being resuspended because of the bioturbation of the amphipods. And a study that looked at that was one by Dyson, and that would increase the availability of algae to these consumers as well. So can I tell if it's facilitation or attraction? I don't really have the ability to do that. But one thing that I do have is those correlation coefficients between the density of meiofauna and chlorophyll A and the density of meiofauna and amphipods. And consistent with facilitation, the meiofauna densities were strongly correlated with amphipod density, but only moderately correlated with microalgal biomass. So if you just look at the correlation coefficient for copepods and nauplii and teneids, it ranged from 0.65 to 0.79 with amphipod density, but it was lower, although significant, for the correlation between the density of copepods and nauplii and teneids with microalgal biomass, 0.51 to 0.56. This kind of makes sense if it's facilitation, because the dense amphipod patches should reduce the algal biomass, even though facilitation could be occurring because there could be more primary production associated with that situation. So I do think this is important information. In other words, the amphipods in this case may have facilitated the recovery of many of the benthic invertebrates in those really severely injured, heavily oiled sites. And of course, if the invertebrates are coming back, then their services, their ecosystem services, would be coming back as well. Copepods, amphipods, and teneids are important fish prey in this location. And then Jaron and Michaela have done a great job of listing the ecosystem services with meiofauna in general. All of those would be enhanced at HV. And then interestingly enough, one group that was enhanced were juvenile bivalves. We know there are lots of adult ribbed mussels in that location, and we know once they reach the adulthood, then they positively affect marsh vegetation. So there's potentially a great importance associated with this. All right, so let me summarize all this in just one slide. For one thing, the full recovery of the meiofauna in those heavily oiled sites is definitely taking longer than a decade. The initial recovery mirrored the return of vegetation and microalgae. But for the longer term, the vegetation cover was slow to recover, and that favored high densities of benthic microalgae, and that did enhance the recovery of some invertebrates. And we think the macroinfauna, the amphipods, could have facilitated it even further. But for some organisms that just recovered slowly, the soil quality seems important, especially density of the soil and living below ground roots and rhizomes. They're slow to recover. And it was slow recovery of the invertebrates as well. So good place to end. I just want to thank the funding sources, the Gulf of Mexico Research Initiative. And although I cited some of my coworkers, we had many other collaborators I didn't have time to mention, research assistants and so forth, that could be associated with, were associated with the project. So I'm going to stop there. I'll leave this up if we want to talk about it. It's just an intention to show you how facilitation might have worked, but be glad to try to answer any questions and so forth. Thank you.